I want to thank you all for cutting your lunch short to listen to me speak. This is my first time to the Mars Society meeting, and I'm embarrassed to say that, but I look forward to continuing attending these meetings. You guys have a lot of energy. What I'm going to talk to you today is about our Mars greenhouses projects that me and my collaborator Ray Buckland from UF have been working on for the last several years, and talk about a project I've been lucky enough to work on the International Space Station called Tropy 1 and Tropy 2, and some of the issues that we had with growing plants in space. Why grow plants on Mars? Well, if we want to have a long-term establishment or colonization on Mars, we're going to need plants for sustainability. We're going to need them for our food. They can be used as fuel source. Fiber, think of all the wipes that you use every day cleaning yourself. You're going to need clean water, and plants can help provide that to humans. They can also provide some of the oxygen requirements, as well as medicinals and sanity. They will keep them from fighting if they're busy planting a garden. And it's a lot of fun. It's one of the largest hobbies in the United States is gardening. So why wouldn't it be one of the largest hobbies on Mars? And to that end, if we were to design a greenhouse on Mars, there's a lot of design considerations that need to be taken into account. And this by no means is an exhaustive list of all of the considerations that need to be taken into account, but some of them include gravitational acceleration. Plants seem to respond similarly to 1/3 gravity as they do to 1g here on Earth, and that's from the studies that I've been doing on the International Space Station, so that's a good thing. It's also good in the sense that things don't weigh as much, so moving things around would be a little bit easier. One of the issues is lower light levels on the surface of Mars, particularly if you have some of those dust storms that were discussed earlier. And you have higher UV levels. So if the materials to make your greenhouse don't filter out some of that UV light, it could cause damage to your crops. The gas phase on Mars is primarily CO2, which is good for plants up to a level, and I'll report some of our findings on that. But it's only 1/100th of the total atmospheric pressure of here on Earth. So that is a challenge for designing a greenhouse. You also have very cold temperatures, so heat transfer issues are going to be a major design constraint. Your structural loads are going to be mainly due to the higher internal pressure of the greenhouse. And dust, the rovers got covered with dust. Luckily, the winds blew that off. Would that happen on a greenhouse? If dust starts to accumulate too much, you're going to have lower light levels, and you'll have to have more supplemental lighting and more power, et cetera, et cetera. So there needs to be a way to mitigate dust on the surface. You also need to design for very precise nutrient and water recycling and handling. I'm not going to talk about that. That has many researchers are working in that area. So there's many concepts of what a greenhouse should look like if it were to be on Mars. And in the top left-hand corner, you have these self-automated domes that are covered in a shell that provides thermal insulation at night. And then during the day, the idea is that these would open up, would reflect the light into the chamber where the plants are located, also have provide solar energy for running the unit, and you'd be able to grow your plant. So they would provide that thermal barrier at night and also direct the light during the day. Other designs have them linked up with the habitation units that may be buried in regolith, and the explorers could potentially go out either suited or directly if the pressure and the atmosphere was conducive to human use, as shown by Ray Wheeler on the Mars greenhouse in the lower corner, be able to go out there in short sleeves and do your hydroponic gardening. Other design concepts include building completely supplemental lighting growth chambers on caves to use the caves as sort of a thermal insulation, and you wouldn't have to deal with as much temperature cycling. Or using fiber optic systems to guide light into buried chambers. So there's a lot of designs, and it depends on when you're going and your particular desire for what you want to do. But the first types of greenhouse are likely going to be these automated, self-contained systems. And they're going to be deployed from unmanned landers that are going to be low mass and can fold up and deploy, and you can grow plants. More of a scientific experiment than for a production unit. So they're going to be very small. They're likely going to be grown at low pressures. And the reason why this is critical is for the structural load on the material. The lower pressures, as Nathan was saying in the earlier talk, require less mass in the structure to maintain the delta P, or the differential pressure between the inside and the outside. But not only that, if you do have a material that gets damaged, higher pressures, you're going to have a higher loss of gas. And gas may be very difficult to get for particular plants. So lower gas, lower total gas is required, lower pressures. So there's been studies since 1963, and this is pretty exhaustive of all the studies that have been done on low pressure growth for plants. And what we do know is that plants need a certain amount of oxygen during certain phases of their growth. For example, they need some oxygen for germination and surely during flowering. So you do have to provide some oxygen to them. But if we drop the pressure down to around 20 kPa, or 1/5 of an atmosphere, plants seem to do just as good as they do under normal atmospheric pressure. If you look at the different types of crops, there's only been a handful of crops that have been studied under low atmospheric pressure. And I'm not including the studies that look at high altitude plants here on Earth, which can go down to 55 kPa. There's been many studies in that area, but they're usually, many of them are not as edible or things that you would consider for food production. At Kennedy Space Center, they actually build one of these conceptual greenhouses, and it's shown there in the left picture. And it's done at 25 kPa, and they enriched it with a high level of CO2 relative to Earth, and supplemented it with 5 kPa of oxygen. And that Mars dome is kind of the concept in the right picture, where you would have these little modules where the explorers, if you could see them, they're suited up, could go out and take one of those modules or move them into the habitat, and you can collect your food from those little domes. Their experiment that they did, they only did one experiment. So it's very sad. They ran out of funding or interest in the area. We're not sure which one followed the other. But they grew lettuce for 45 days, and I want to make clear, though, they germinated the seedlings at 101 kPa. So they germinated them in the laboratory setting and then moved them into the low-pressure environment at 25 kPa. What they found was that the plants did just fine. They had a typical weight accumulation as the 101 kPa. So this gave hope that, you know, this is doable in those type of dome setups. We at the University of Florida, in collaboration with the University of Central Florida, 4 Frontiers, through the Florida Space Grant Initiative, we're funded with a Ralph Steckler Opportunity Grant, Phase 1. Unfortunately, I can't report we got Phase 2 because that was given to colleagues at University of Arizona, but I don't want to digress on that. But I'm going to talk about our Phase 1 project because it was, gave us some very interesting results. We were looking at how this low-pressure environment affects plants, and can we start to develop decision support tools, as Nathan talked about in his earlier presentation. You need to be able to predict how to manage these crops if something goes wrong. How much water are you going to need? What happens if this occurs? Can you design engineering tools to do that, similar to the crop models that are used here on Earth? So other issues that we're looking at is mass transport limitations. Because you're under lower atmospheric pressures, you have more difficulty moving things around and maintaining mass transport. And we're looking at identifying materials that would be used in the construction of these inflatable type greenhouses, and whether or not they would survive Mars surface conditions. And lastly, but not least, is portion of our project was to develop innovative educational and outreach for K through 12. This is to get them excited about space research, space exploration, because they're the ones that are going to be doing a lot of it, hopefully. And they're also the ones that will come up with some great ideas that we haven't even thought of. So we built these. Our idea was we need to build small units, prototypes, to be able to study how plants grow here on Earth under low pressure, and then see how we can improve some of the models for Earth crops and apply them to Mars agriculture. And we're also developing new sensors for detecting dissolved oxygen, for example, very tiny sensors, and different pH sensors, et cetera, that are very small. But I'm not going to talk about our sensor technology in this presentation. So one of PhD student Aaron Wilkerson, now at Oak Ridge National Laboratory, worked with Ray on building 3 prototype Mars greenhouses at Kennedy Space Center. She did her project there. And she basically had 3 glass bell jars that had lots of sensors packed into the bottom, and they include oxygen, CO2, humidity, temperature, light, etc. So she was able to monitor the environment very closely, both where the plant was located and down in the housing unit. And she was able to grow plants in these small chambers. The issue was she had very small chambers, but some of her studies included looking at different pressures. She used 4 different pressure treatments, 2 different carbon dioxide treatments, and 2 different PAR, photosynthetic active radiation treatments with 340 and 160 micrometer squared per second. So she did a variety of studies on radish. So she took radish plants and grew them in pots at normal atmospheric pressure, or Earth sea level, and grew them to about 24 days old. Then she moved them into these low pressure chambers, acclimated them for 3 hours to the low pressure conditions, and then provided this treatment and monitored the evapotranspiration. And I want to clarify, she's measuring evapotranspiration, which includes the transpiration or water loss from the plants, as well as the water loss from the soil. So it's just a note, because later we look at transpiration. What she found, and the points represent individual plants and their evapotranspiration rates, as you lower the pressure, you have increased evapotranspiration. This is not very surprising, because you have enhanced diffusion as you decrease pressure. Molecules can move around a lot faster. If you increase the CO2 from 40 PA to 150 PA, You can reduce that evapotranspiration, and that's mainly plant, due to the stomata, or the pores on the surface of the leaf, will close during elevation of CO2. So she could lower the evapotranspiration by increasing the CO2. And the lines there are her models that she used based on Penman-Monteith crop models for evapotranspiration, and they followed her data pretty well. She also looked at what the effect of different light conditions would do on this evapotranspiration. Now, the stomata are very light sensitive, so when morning starts coming, plants open up their stomata, start to get CO2 in and photosynthesize. If you increase the light, they may even open up a little bit more to take advantage of that CO2, especially in conditions with low CO2, and that's at 40 Pa is Earth atmospheric CO2 concentration. So when she increased the light levels, she had enhanced evapotranspiration, but not at the very low pressures down at 20 kPa. So regardless of what the light treatment was, the evapotranspiration rates were the same at this very low pressure. She also looked at engineering issues that are important under low pressure systems, and she's looking at convective heat transfer under low pressure in a simulated leaf. It's a aluminum foil with thermocouples underneath, and she has a little fan, computer fan, blowing across it to measure the heat, rate of heat loss on the leaf surface under the different conditions, or the different pressures and different velocities of air. Not surprising, as you drop the pressure down, the rate of sensible heat transfer drops. When can this be a problem? If the leaf temperature is heating up and you're using fans like they do in greenhouses to get air moving around, you don't have very much air to move around. So you're not going to be able to mitigate some of those high temperature areas within the greenhouse. So it's a very, very challenging issue. Aaron's experiments could contain one plant per chamber, or 2 plants in one pot, but basically 1 pot per chamber. They were also made out of glass, which isn't very safe when you start changing pressure, but we wanted to build a little bit more sophisticated and bigger chamber to be able to do multiple plants, and that was done by Hemant Gohil. He recently got his PhD and now is at California State. So we built up bigger chambers with similar types of controls as Aaron's and looked at 4 different experiments. One was to see how low of a partial pressure of oxygen were required for the germination of radish. If you were to take plants from high pressure to low pressure, what effect does that have on them? And you can think of that as possibly some greenhouses may be connected to the habitation units on Mars, so they may be at higher pressure, and then you may have some further down the road that would be under lower pressure, similar to seedling greenhouses, and then the plants are moved here on Earth. So you may have different greenhouses for different purposes or different developmental stages. So what happens when you transfer plants from high to low pressure? What happens when you increase the CO2? And long-term and short-term effects. Aaron just studied daily measurements. Hemant was going to look at germinating and growing plants in low pressure. What he found for partial pressure of oxygen under 33 kPa, we picked 1/3 of an atmosphere as our target low pressure treatment. He found that if you drop the partial pressure of oxygen below 10 kPa, you ended up with a significant reduction in the percentage of seeds that germinated. So you've got to balance how valuable is oxygen versus how many seeds. Of course, you want 100% germination. So going about 10 kPa, there was no statistical difference between the 21 kPa, which is found here on Earth, and the 10 kPa. treatment. So
101 is our atmosphere?
yes, 101 is, I'm sorry, the far bar is just plants growing in a chamber that's at 101. The next experiment he was doing, which wasn't what we wanted to do, but we thought, let's germinate the seeds and then bring them into the low pressure environment. So we germinated them from seed at normal pressure because you want to make sure all your plants are uniform and they're germinated. And we transferred him at 1 week. But every time he did that, he did it probably a half a dozen times or more, he ended up with plants that looked like that in the left-hand corner, the top panel. They could not keep up with the transpiration rates when you transfer from 101 to 33 kPa at 1 week time frame. You could germinate seeds in low pressure and grow the plant just fine. You couldn't transfer them at 1 week. The reason for this, we believe, of course we have to do more studies to determine that, is because at 1 week, the main leaf surface is from cotyledon leaves, and the root system is very immature at 1 week. You have very little root system. So when you transfer it into the low pressure, these cotyledon leaves are transpiring, the roots can't keep up, and you end up with a drought response. And that's what our hypothesis is. So you have to be careful when you move from higher to lower pressure. So if you were to grow plants for a crop in low pressure, for radish, we found that there was no difference in biomass, dry or fresh weight, or the root to shoot ratio. So they were very similar in that regards to plants grown at normal pressure or very short acclimation periods. However, what we did find was that the leaves were much smaller and thicker. And this is not surprising because under low CO2 you have enhanced, under low pressure you have enhanced CO2 diffusion into the leaf surface. And leaves will become thicker under higher CO2 conditions. So that was interesting. But what we also saw was that our transpiration rates and our CO2 assimilation rates were higher under the low pressure treatments. So now, somebody brought up a question, what atmosphere, why wouldn't you just pressurize with CO2? And plants love CO2, right? They'll just do just great. Up to a point. And we wanted to see how much CO2, what effect that would have on the growth and development as well as the transpiration and CO2 assimilation. But I just have a picture here showing increasing the partial pressure of CO2 at a 33 kPa or low pressure chamber. We ended up with plants that did not look great or very delicious. We had this redding color on the leaf. So if you were to pressurize with CO2, and Ray Wheeler from Kennedy Space Center has done studies on this as well, there's a certain limit to CO2 that plants will tolerate. Then they start to have starch accumulation, senescence, different stress responses. So you have to understand your crop, and different crops are differently sensitive to different gases. So each crop would have to be studied that you would want to grow on a Mars colony. The third experiment I'd like to present is probably the most interesting from an engineering standpoint, and this was done by Inka Hublitz under Ray. And what this is a chamber that mimics the actual conditions that a Mars greenhouse would have. So what it is a dome that is under higher pressure than the surrounding environment. And what it is housed within a vacuum chamber. So that dome, you'll note the bolts are to keep the top from blowing off. The chamber was run at 25 kPa, and the outer chamber, I believe, was at 4 kPa. We couldn't get it down to the 1 kPa of Mars, but anyway, she wasn't able to get it down to exactly Martian conditions. And the chamber itself was housed within a commercial freezer of minus 30 degrees C. So she was able to get a very cold environment with low pressure and put her chamber in there. And what she did a very short-term study, 7 days with lettuce. She found that the lettuce did just fine during that treatment. The actual chamber itself, though, had two zones. One was where the plants were located. She used her stovetop heater to heat around where the plants were located. So you know the coils that you get from your stove, she pulled it out and plugged it in and put her plants around it to keep them warm. But in the lower part of the chamber, it was cooler because there was a separation device. So all the water that transpired from the plants accumulated in the lower portion of the chamber, and you ended up with chunks of ice. So you've got to be very careful of microclimates within any chamber or greenhouse that you would build on Mars. And that's the industrial food freezer that she put her vacuum chamber in. And here's how they looked. They didn't show any signs of cold shock, heat shock. They grew just fine, similarly to what were found under laboratory conditions. So in summary, if you want to grow plants on Mars, you can germinate them, but they have to have enough partial pressure of oxygen, and this is going to be species dependent, so different plants will require different amounts of oxygen. You're likely going to have increased transpiration rates. They seem to do okay with this, provided that you have a system that provides enough water. And if you go too high on the CO2 level, you may end up with starch accumulation in the leaves or leaf damage. So not likely going to pressurize the vegetable with the CO2. If you transfer your seedlings from high pressure to low pressure during the cotyledon stage, you have to be careful because you may end up with what we saw were wilting, and basically the plants ended up dying. And the microclimates within the Mars greenhouses need to be carefully engineered to prevent what we were seeing, which was the chunks of ice building up in different locations. In addition to looking at plant growth, we're also looking at the types of materials that would be used to build a Mars greenhouse. And we used the Mars simulation chamber at Kennedy Space Center to look at the effects of the Mars environment on different plastic materials. And this study was just very preliminary to develop our techniques on how we would treat these plastic materials. These materials would be used perhaps in the construction of an inflatable or as coatings on the surface of maybe glass to prevent glass from shattering, et cetera, if you were to go with a glass-type greenhouse built on Mars. We took 5 materials. We did 42 days of Mars treatments, and we measured the transmittance and the tensile strength of the materials to determine if the Mars environment caused them to break down. What we found, we just threw in 5 general plastic materials as a starting point, and what we found was all of them showed a reduction in transmittance in the photosynthetic active radiation region. So they wouldn't be very good candidates for some of these deployables. Now you can put coatings on the surface of these to reduce UV, and that's our next studies that we'll be doing in the coming year. If we looked at the yield stress of these different materials, what we found were 2 of the materials, polycarbonate and cast acrylic, had a reduced yield stress after exposure to the Mars condition, and just indicated by the 2 stars showing those 2. The rest behave similarly statistically. As the final part of this GROW project, we were developing education and outreach tools, and I'm sorry to say that Joe Palea won't be speaking tomorrow, but he's developed, along with us, interactive tools for students to go in and learn more about Mars exploration. It's on his Crazy for Mars website. And he's put some new lesson plans on there on what it would be like to grow plants on Mars, and has an interactive video of some of the time-lapse photography we have for growing plants. We also developed some of those models for the computational biology workshop that I was an instructor at, to understand how pressure affects transpiration and photosynthesis. So students could go in and lower the pressure and see what happens to transpiration. We had a variety of students that have worked on the project. Samantha Smith, she did some of the plant studies and presented at the American Society of Gravitational and Space Biology. One year, she got the second place poster award, but the following year, she worked on more of the materials and was able to get first place. So it was very rare that a student ends up with 2 awards back to back. In the interest of time, and I want to move on to my space station project, just to give you an idea of some of the issues that you might face for Mars colonies. I'm going to just briefly say there's other folks working in Mars greenhouse studies, as Nathan presented earlier. University of Arizona, they're not doing low pressure studies, they're using normal atmospheric pressure and have a deployable greenhouse. And Ron Lacey and Fred Davies, unfortunately, this research is winding down at Texas A&M. And Mike Dixon at Guelph in Canada has some low pressure chambers that they're also doing some studies on. Okay, so in overall summary, you need to be able to control the gas phase very carefully. Make sure your temperature control is maintained, or you're going to end up with ice chunks. There's only a few handful of crops that have been studied in low pressure, so there needs to be more focus on the different types that would be used, the abiotic and biotic stress, and maybe we can grow specialized plants for Mars. Maybe we haven't developed the right plants. And we need to improve some of the technology to control and monitor the environment. So briefly, I want to talk about a project that I was involved in called Tropy 1, and it ended up being reflown on a second mission called Tropy 2. Little different science between the two, but it was run by John Kiss from Miami University, and I have to say this is Miami University of Ohio, not Miami University, or University of Miami in Florida. It was also supported by ESA, NASA, and the University of Florida through some startup money. Before I go into the space experiment, I'm going to give you a quick biology 101, talking about the power of movement of plants from Darwin. Plants respond to their environment in a variety of different ways. One of the ways they do this is called directional growth in response to a stimuli called tropisms. And you've seen this in every day, any of you that's ever grown a plant. One such tropism is called thigmotropism, and that's directed growth in response to touch. So this plant's winding its tendril around another plant to grow up. We have phototropism, the directed growth in response to light. We have gravitropism, the directed response of growth in response to the gravitational acceleration vector. Here on Earth, you constantly have phototropism and gravitropism interacting. So it's very difficult to separate the two of those out. If you want to study gravitropism on Earth without interacting effects of light, you just turn off the light. If you want to study phototropism without the interacting effects of gravity, you can't do it. Or there's very difficult, you have to do drop towers and things like that. So International Space Station provides an environment to study the effects of photoresponses without dealing with gravity or microgravity. But you can ask a whole lot of other questions while you're doing a project on the International Space Station, like how do they grow on space versus ground? What other tropisms are coming on? If we're looking at phototropism, we may see some other tropisms. And how do these types of things interact is our main focus. The hardware that we were using to grow the plants was the European Modular Cultivation System from ESA. And what it is a incubation chamber that contains 2 centrifuges. And that's great because you can have one centrifuge giving a gravitational acceleration and the other one providing microgravity. And you as a PI can go in and propose to do the experiment in these ECs, which are called experimental chambers, that are about 1 foot long and 3 inches deep, 3 by 3, and design hardware for your particular experiment. They will provide water, light, gases, temperature control, and video or digital images to you. In our case, we didn't use either the water or the lighting system. We had our own, as I'll show in just a minute. But interesting to note, this cultivation system was designed for plants, but they've ended up using it for fruit flies. So what's good for plants apparently is good for fruit flies. So it's interesting that the hardware is being cross-disciplined. We designed these small chambers. We were growing Arabidopsis seedlings, which are the model organism for plants that are, the genome has been sequenced at the seedling stage, so they're very tiny. The grid lines on that upper block with the seedlings are 3 millimeters, so we're talking very tiny seedlings, small plants in space. Space is limited in space, so the smaller the plant, the better. And we were treating them with different light levels. The actual cassettes that they were grown in are pretty complex. They have a little water delivery mechanism. They have a clear plastic coating on the surface to prevent condensation from occurring to maintain image integrity. They have different LEDs in different locations, etc. They also have holes for gas exchange in the system, and they also use technology developed by Fresh Express where they have gas permeable membranes in the system. This is the final configuration of the EC, or experimental chamber, with 5 cassettes. Each cassette had 14 seedlings. And the 2 images are light from above and light from the side. And what we sent up were dry seeds that were sewn on filter paper on these cassettes. And it went up with the hardware on our first Tropy 1. During the implementation of the experiment, the astronauts loaded up the ECs into the EMCS, and we performed the experiment with downlinks to be able to provide feedback if we needed to run part of the experiment longer. As I mentioned, we set up dry seeds. What some of you may not know is that many seeds require red light in order to germinate. And so we hit them with a blast of red light to synchronize germination. We then grew them all under the 1G centrifuge with the white lights to get nice straight seedlings. And then further down the experiment, we turned on the side lighting, whether they were red or blue, and looked at different gravitation levels to see how the plants curved in response to the different light stimulus. At the end of the experiment, the astronauts came in, opened up the EC, opened up the EMCS, took out the ECs, and put our hardware in the freezer so we could do genetic analysis on the RNA. And then we got them back at some point. This is the actual timeline, so it's showing different downlinking and different processes that were done at different time points. They were only run for about 6 days. At the end, we have videotapes and images to analyze, and we have the frozen samples to do some gene expression studies. The differences between the two experiments: Tropy 1 was launched in 2006, on July, and the last experiment wasn't performed until December. So there was quite some time up on the space station that these seeds were sitting on the hardware, and it was a year later, or half a year later, that we ended up getting all of our seedlings back. For Tropy 2, it was launched in February. We were getting it back in April. So it was only a 2-month, roughly, time frame for that experiment. So if we look at the images from the two different experiments, I don't know if you can tell, but the seedlings in Tropy 1 look really pathetic compared to the seedlings in Tropy 2. And the white lines are actually the seedlings. And we found that we had very poor germination in the experiments for Tropy 1, but very good germination in Tropy 2, similar to Earth. This is a close-up image just showing the two experiments, and the little bar graph showing the percent germination in our Tropy 1 experiments, very poor germination compared to our Tropy 2 experiments. That little circle highlighted in the Tropy 2 is showing image degradation on the EMCS. Since these experiments were performed 4 years apart, we were able to compare images from 2006 with 2010 and could actually document some of the image degradation in the hardware. So that was one of the neat things about looking at that, probably from space radiation. What happened on Tropy 1? Well, we had very poor hydration. Not enough water was making it to the filter paper where the seeds were located. And if you see on the top cassette, there's lines. That indicates good hydration. They start to appear when the water hits the membrane. If you look at the middle cassette, you don't see those lines. Some of our cassettes did not even get hydrated. So that ended up in a loss of science. What also happened on Tropy 1 was the astronauts took out the cassettes from the Melfi and let them sit for over half an hour before putting them into the cold bags that would be returning to Earth. That causes the warming up of the cassettes and degradation of our RNA, which, for those of you that are familiar with gel electrophoresis, this is showing RNA being degraded in the Tropy 1 experiment, which is just there's nothing up here. And in our Tropy 2, we ended up with very good bands that indicated good quality RNA from that. Not all of our Tropy 1 RNA was bad, but a good percentage ended up being lost. So we took the RNA that we had from Tropy 1 and ran microarrays on them and compared them to Tropy 2. And what we found were that many stress-related proteins were differentially regulated on Tropy 1, meaning plants were under stress. That's not surprising. You could tell by the images they were under stress, but now we have a genetic backup to say exactly what kinds of stress, and they were drought stress responsive. With Tropy 2, we're getting really good science. We're starting to look at some of the genes that are involved in cell wall development. So the different gravitational accelerations show very different cell wall genes being differentially regulated, suggesting further avenues to study in cell wall development, fundamental science. So the challenges that we encountered in the first experiment, we were the first people to use the EMCS, which was great in the sense we got a lot of engineers working with us, but it was bad in the sense that we were the ones to test the hardware and find out all the little glitches We had some problems with image calibration. Some of our images were of the wall of the cassette. We had some power outages that ended up, luckily at time points where it didn't affect our experiment, but it's difficult being the first. Sometimes it's a good thing, sometimes it's a bad thing. We ended up with hours of useless video, and I'm saying video because we got 8mm video tape back from the space station. Now they just do digital imaging, which we were pushing for many, many years ago. So much easier analysis. And we ended up with much better RNA or genetic material to look at in the second experiment. These experiments descriptions are found on the NASA website, as well as we've published this year in the Advances in Space Research. And it goes into much more detail of some of the other issues that we found. This work, this is not an all-inclusive list of all the people involved, but I particularly want to acknowledge John Kiss, who's the PI on the Tropy project. I've mentioned Ray Buckland. He's been working with me since I arrived at University of Florida. We've brought on some new faculty to look at the sensor technology, and we've had, this is just UF students, but we've had other students from other universities participating on this project as well. Oh, and our funding. Our funding for the Tropy experiments was provided by NASA. We had the Ralph Steckler grant for our Mars greenhouse, and the university scholars provided money for undergraduates to do research, and the Florida Space Grant Consortium has been giving us grants to keep us alive for research. So if you have any questions about growing plants on Mars and or in space, I'd be happy to, okay.
Hello, I'm Rick Smith. From what I've taken from your talk, it would not do to simply pressurize Mars's atmosphere. You'd have to add oxygen so it's able to germinate, and there'd be too much carbon dioxide. So
correct.
You'll need a special mix. My second question is, Robert Zubrin and others have said that using Martian carbon dioxide, you could make simple plastics. I think something useful would be, what is the simplest plastic that people on Mars could create that would be resistant in that environment?
That's an excellent point, and we are working with Joe Palea, who's helping us to focus on that part of the project. We're also looking at innovative technology that would be useful here on Earth, because that's where our funding comes from. So for example, USDA. But you're absolutely right. We would want to look at materials that could be made and built for Mars.
Do you have any idea what would be the simplest plastic that would be reasonably resistant?
Ooh, I do not know that. I'm sorry. But maybe somebody out here might be able to.
In the chamber that was surrounded by the cold box.
Yes.
The chamber was pressurized. Did the researcher make any attempt to conform the PAR spectrum inside the chamber to that you would expect on Mars?
No, unfortunately we didn't. With good PhD students, they tend to find jobs and leave. So we didn't get to that level to look at actual light. And it's very difficult to find good data of what the light PAR region is on the surface of Mars. Okay. So if anyone has some good...
We should talk.
Yeah. We're looking for a good analog.
Have you had an opportunity to look at plants that may be a little more adapted to the Martian environment, such as grown at high altitude locations around the world, like Tibet or Peru?
We have not looked at that. One of the reasons is they tend to have a little longer cycle for producing. And we want to get experiments going fairly short time because you want to do many replicates and time is worth money. So we haven't looked at that, but it is definitely something that could be explored further, is looking at high altitude plants and how they adapt and respond.
It'd be a little more UV tolerant too.
And more UV, absolutely.
So what, for like PSI and stuff, I forgot how to convert it. What levels of carbon dioxide look like they're beneficial? Because they grow faster and low, right?
Mm-hmm.
About what level looks beneficial, and where does it start to become a problem?
Okay, this is going to be very plant specific because different plants respond very differently to different concentrations. So I'm putting that caveat up front. But for radish, if we go 4 times Earth,
Normal Earth?
normal
Yeah.
Earth, we start to see some problems.
Do you think higher water levels might help?
No, these were hydroponically grown. I didn't
Oh,
state
okay,
that.
so they had plenty of
They
water.
had plenty of water. And so transpiration was not an issue as far as not enough water availability.
And you don't know about light levels?
I'm sorry?
And you weren't working with different light levels?
Just, we looked at a couple different light levels, right? But not Martian levels.
Thinking back to the presentation this morning, assumptions and hidden costs. We have a built-in assumption, which is low pressure. Why? Because we save on mass. But hidden costs. In watching your presentation, I'm seeing lots of hidden costs. So is low pressure necessarily a good answer? Maybe going full pressure might be a better answer. So my question to you is, has somebody developed a figure of merit for greenhouses that would include human labor, obviously energy, mass, and obviously output is what you care about, edible food at the end of the day?
Yes, NASA's looked at this. Ray Wheeler is a co-author on some of the documentation for that. And I can probably send you that information as far as costs and equivalent system masses and to grow plants in space and or Mars. We have tried to work with NASA to see what level pressure they're likely to have for human habitation. And so they're looking at about 55 kPa, at least that was the last tidbit of information we were getting. So 33 kPa, and we were looking at 66, which I didn't show the results. It's probably going to be lower pressures.
The plants that you worked with, the radish and the lettuce, they never got to the point of flowering or setting seed. Has the Arabidopsis work said anything about the ability of plants to reproduce under low ambient pressure conditions?
That's an excellent point. And flowering has not been studied, as far as I know, under low pressure conditions, other than the high altitude studies that have been done on Earth. That's been looked at. So, no.
Are there any indications from the high altitude studies that would give you a means to guess what might happen at low pressure?
My guess from looking at it is you have to have plants that are adapted to lower oxygen levels because oxygen is also very critical during flowering time. So that could be one of the areas in flowering that needs to be further explored under low pressure conditions.
Thank you.
Mm-hmm.
Hi, so I know you were saying probably because of the newness of this area of research, it's only been happening for for a few decades and only really a lot for the last 15, 20 years or something, right?
Well, since 1963, they've been studying plants in low pressure, and maybe the Russians have been doing it even longer. I don't know.
But there's still not a lot of data.
There's not a lot of experiments that have done that.
So if in an attempt to fill the data gaps, if you wanted to do experiments to find, to study one aspect for a bunch of different plant types, what aspect would you want to know for like the hundreds of plant types or dozens even? They didn't have that much money.
That happens to be a loaded question because it's whatever your little area of research ends up hanging your hat. Some people are interested in flowering time, some people are interested in germination. All of these are going to be very important. So I can't designate the biggest area that needs to be focused on.
It sounded like you were interested in optimal CO2 pressures for low pressure. That makes sense.
Yes, we were interested in because you would think to use CO2, it's there, why don't we increase it and use that as a mechanism for pressurizing it?
Okay.
But that's where our interest lied.
Yeah. And one last question. Has anyone done any research on how dehydrates fare in low pressure? I mean, like, I just think for some reason, it'd probably be nice to have a plant that if, for whatever reason, you couldn't give it water this week, it could die and then come back to life later.
Yeah,
So-
like there's a prayer plant that can dehydrate and get into this little ball dried mass and could be like that for years. And you add water and then it just, terrestrial studies are working very hard on looking at some drought-tolerant species. So
Okay.
NSF and USDA are funding a lot of research along that line.
Okay.
So I think there'll be some interesting plants out there in the future.
Okay. Thank you.
Okay, the last question.
I have a quick non-scientific question.
Mhm.
The radishes and the lettuce that you grew, did anybody taste them to compare with the normal?
The PhD student that worked with the radish in the second does not like radish. But I tasted them. They tasted perfectly fine to me. He tasted them, but he said, I don't like them anyway, so I don't know what a good radish would taste like. But the lettuce, no, I don't think Aaron ate any of the lettuce. Thank you.
And there was discussions in Australia about plant growth as well and gravity issues with that. And he's from Russia, so he knows all the research that they're doing for humans to Mars there. And he kept making the point that there's no issues with gravity in plant life. The only issues is with bone loss, things like that. So he kept saying over and over again, actually he repeated it, plant growth will be fine, plant growth will be fine. It's just humans life that's going to have the issue.
Plants will orient to light. So there's actually a mutant, Arabidopsis, that doesn't respond to gravity. And you can actually grow it horizontally with light on one side. So it's true that they'll grow. I mean, the topsy-turvy tomato, everybody's seen that where it's growing down. So gravity probably won't be a problem, except for natural convection. That's going to be an issue. And Mary Musgrave has studied that extensively, looking at internal pea pods. Because there's no natural convection occurring under microgravity, the pea matures at different rates. So you get these pea pods that are brown on one side, green on the other. And you can't put little fans inside a pea pod. So.
I just came back from Australia, a NASA Spaceward Bound trip, where we brought the Mars suit out to Australia with us in the outback, and we did some testing with it. So I have some pictures with me, and I have that I'll share with you. We did some education outreach, which is my background, with some Aboriginal children out in Australia. So you will see pictures that we currently took of the Mars suit, and also. And then I'll talk about the collaboration we've been doing with ESA, the Mars 500. The Mars Desert Research Station crew created a video and a communication between MDRS this season and the Mars 500. And then I just mentioned I also spoke with Vadim Gushin in Australia. He was very excited about the Mars Society collaborating with the people that are in there for 500 days and said, this is really excellent work for their mental health, this collaboration between people who have experience, you know, even though it's not 500 days, it's only 2 weeks that we have our crew rotations at MDRS, but they still have a little bit of an idea of what it's like to be in this sort of situation. And so he said he really appreciates that we collaborated with them and that he wants more collaboration between the Mars Society and the Mars 500 crew before they get out in November of this year. So he actually invited me to come to do a tour of IMBP, IMBP, I believe it's called, and also where they're stationed, and also bring some MDRS crew members out there to continue some communication between the two groups. Okay, so I'm going to share with you, this is and is it pictures? Okay. And I need to change it to a certain time. So that's can you forward it? I don't know if I have probably 26 seconds. This is a video of a news broadcast in Australia that they covered about this NASA Spaceward Bound project that I was on that mentioned the Mars spacesuit. And yeah, at 26. That's not the spacesuit. That's not the spacesuit. Okay, just get sound. I can't see anything. Okay. Getting the sound ready for you.
NASA scientists have landed in WA to bid to unlock the secrets of Mars. They say the Pilbara could help pave the way for a manned mission to the Red Planet.
NASA's on a mission to Mars via Marble Bar in Western Australia, suiting up for space in the outback. But this is no science fiction.
The exploration we're doing here is part of the science motivation of this whole story.
7 minutes. 7.
The colour's not all that's in common with the red planet. Fossils here date back 3.5 billion years, the sorts of microbes that could inhabit other parts of the solar system.
We're doing here in the Pilbara what I'm hoping to do on Mars, which is find evidence for early life on a planet.
Part of the agency's Spaceward Bound program, the team's testing a prototype survival suit, more flexible than the regular astronaut outfit, designed for longer forays across the Martian surface.
You need a suit that you can put on every day like you would a pair of old blue jeans and wear them 8 hours, go out and do your work.
Even in an Aussie winter, it's hot work, but that won't be such a challenge on Mars, where it's 60 degrees below zero. Would you like to go to Mars yourself?
It may be a one-way trip, so in answering that, probably no.
Young locals cautious too.
It'd be pretty cold. We've been to Sweden and we thought that was cold enough, so.
While the Pilbara is giving us some idea of what we might expect to find on the red planet, scientists working here are now linking up with Russian experts, examining the effect on humans of a possible 4-year mission to Mars. Russia's Mars 500 project's well advanced, testing how a group of astronauts would handle 18 months of isolation. But the timing for the next giant leap for mankind is still out there.
I'd like to be around to see it happen, but I'm happy knowing that it is going to happen.
Nick Way, 10 News.
NASA scientists have landed in WA in a bid to unlock the secrets of Mars. They say the Pilbara could...
So that's where I just came back from just about a week ago. And I have my office just down the hall from Dr. Chris McKay. I work with him at NASA Ames, and I'm a point of contact for the Mars Desert Research Station and NASA collaboration working together. So that's also something else that I do, aside from being the executive director of this wonderful organization. I would like to show the pictures. So I'm going to show you some pictures of some of the outreach that we did and some of the works that we did. We got to an opportunity to look at 3.5 billion-year-old stromatolites in Western Australia, one of the few areas in the world where you can see an outcrop. And of course, you know, this is the early signs of life. I was actually there to help survey this geologic outcrop. It's going to be called a Dawn of Life Trail. They want to make it available to the public to share with them this wonderful treasure that they have. and also to preserve it and protect it so people don't remove pieces of it. And I'll show you. This is the area that we were working in. There's lots of, the grass is not soft. It's called spinifex, and it rips up your ankles. And this is us on full gear just walking up the hill, going to look at an outcrop. This is a close-up of a stromatolite, a fossilite. This is a 3.5 billion-year-old fossilized stromatolite. And you look at the laminations and looking for cone structures. And basically, these are the reasons why we have an atmosphere, a balanced atmosphere. They released a lot of oxygen. These are the early signs of cyanobacteria algae that use photosynthesis and help stabilize our atmosphere here on Earth. And I just put this in so you can see the scale. Okay, great. This is just a picture. I work with, so it's wonderful that I'm able to be at NASA Ames, and I get to talk with Dr. McKay and Dr. Carol Stoker, and this is actually the first time that they ever went on an outing together, so I had to get a picture of them for, they've known each other since graduate school at the University of Colorado at Boulder, but had never gone out on an expedition together, so I snapped that photo for them to remember it. It's not going.
You messed it up.
I messed it up, apparently. Sorry. Okay, we're going to try something else. Just another picture. There's actually one of the Aboriginal students right there. And if you notice, we're in full gear. We've got hiking boots on. She came in a pair of flip-flops. And she's walking up this heavy terrain with rock, and she actually decided, I'm going to take off my flip-flops and walk directly on these jagged rocks. And this stuff feels like the needles on a cactus. So they're just acclimated to the area and have no problems walking in that environment. So there's a close-up. I was just in awe of her walking barefoot on this area, and we have hiking boots on. So here's a picture of the Mars spacesuit. We were doing tests of timing of them in the suit, and then walking from a certain point to the stromatolite outcrop, and if they can point out stromatolites, then we timed them, and we tested different individuals in the suit to see how they fared. And that's actually that's John Rask, who we were expecting to speak today. He works with Pablo de Leon at the University of North Dakota on this Mars spacesuit. Just some more pictures. This is one of the children. I actually bumped into a teacher who volunteered for this. It's during their winter break. It was winter in Australia while I was there, which is why my voice is a little scratchy, because I think I got a cold. But she was able to bus out a group of Aboriginal children in this area called Nullagine, right near Marble Bar where our outcrop was, and she brought them out there to witness what we were doing. Just some more pictures. And that's me taking a picture with evidence that I was there. Here's. One of the kids actually, they saw the Mars suit, and but they were more interested in my camera, so they took off with my camera and started taking pictures of things with it. So this is one of them. And of course, we had to get a group shot of them. This is the reason why we do Space Rootbound. It's to put teachers and scientists together in the field, and basically it's so that teachers can go back to the classroom and inspire future field scientists, Mars analog field scientists, other planet field scientists. Next. Here's one of someone getting out of a suit, and it was winter in Australia, but it was still too hot to put on. There's usually a blue insulator that goes over this part of the suit, so it doesn't normally look this color. It's normally blue, and they've tested it in the Arctic, and that's where they'll use the outer layers to insulate them. But it's definitely not made for even the Australian winter climate. She was fully just covered in sweat. And the inside lining is rubber, so you basically get out of it and you're in a pool of sweat on there. So the kids really liked our camera equipment more than they seemed to be interested in the suit. And we passed out some NASA stickers, so they started to put them all over. their places. And they're very happy and bright, and it was a wonderful, most amazing opportunity for me to interact with them and to share some of the work that we do for Mars outreach, Humans to Mars outreach and research. And that's me. We got to, we didn't get to swim with the dolphins, but which you could do in this area. This is called Shark Bay, right off the coast of Western Australia. But we didn't get out there early enough, so instead I swam with the stromatolites. Yeah, well, these are 7,000-year-old stromatolites, and it's a protected site that they only allow scientists, researchers to go to, so we were very lucky to have that opportunity. So, okay, sorry about that. I was hoping to show you the video that we had. So Mars Society and our crew from MDRS had actually communicated during crew rotations with Mars 500, and I was hoping to show you the video, the first video that was Carol Stoker's team. She was doing a drilling on other planetary bodies, and they created this wonderful video and sent it to Mars 500, and then the Mars 500 sent one back, but I don't seem to have those downloaded. But what I do have is an additional video that the Mars 500 crew did recently, just in the last week, and sent it to us to show it to you at this convention, so we can play that one. That's that one right there. Yeah.
Dear attendees of the 14th...
Just get it bigger for you. It's all on the screen.
The Mars Society convention. Usually I have the chance of speaking to different sorts of audiences about the Mars 500 experience, but this time it is a pleasure to address an audience that, just like us, shares the belief that we are a spacefaring species and that we should be exploring and expanding our frontiers beyond our wonderful but small, in a cosmic scale, Earth. I am a crew member of the European Space Agency at the Mars 500 mission that aims at gaining better understanding of the issues that derive from the extremely prolonged isolation necessary for a trip to Mars. In here, we're constantly working on experiments in physiology, in psychology, and operations. In a Mars mission context, we are completely isolated from the world in 3 pressurized modules that are located outside of Moscow at the Institute of Biomedical Problems. As of today, we have been enclosed here in isolation during more than 400 days out of a total of 520. It takes not only a very professional, dedicated, and strong-minded crew to go through this, but above all, a crew that is united, that is calm, that is flexible and tolerant in order to face the conditions of confinement. On February, we simulated a stay on Mars, where 3 of us went to the simulated surface and executed field work with several space-rated tools. It was a very exciting period, and although it wasn't a real Mars landing, it was just as exciting. Folks like us that had been enduring sensory deprivation for so long, and that had been building up this excitement on the route to Mars. It could have been otherwise. We were extremely enthusiastic, and it was really a fantastic, I think, and almost mystical moment. After this experience, I really really can't wait for humans to really step on Mars. The second part of the trip likely hasn't been the easiest of them all. Earth seems already like this very strange place. I think it would be very alien-like for us, at least for the few days. It is not the easiest of the moments, but we are very enthusiastic still, and we are driven by the will of seeing the people that we love on Earth. My stay at the Mars Desert Research Station was a great couple of weeks. It gave me the perspective, a different perspective, in order to enrich this experiment with that experience, and I'm very thankful because of that with the Mars Society. And I hope that the Mars Analog Research Stations program will continue providing all this knowledge to all the generations of explorers, of course, until we reach finally the red planet, which we hope won't be too far in time from now. This is my colleague Roman, who will talk a little bit about our scientific program.
Hello, so my name is Roman. I am the second European crew member selected by the European Space Agency, along with Diego. And here I will talk a little bit more about our experiments in the Mars 500 facility. So we have roughly 100 experiments, which are divided in 5 big groups. The first one is psychological experiments, medical experiments, physiological experiments, hygiene experiments, and operational experiments. So here we are in the gym of our modules, and I will talk a little bit more about the physiological experiments because they are directly related with sports and other experiments that we can see just here. So regarding sports, our crew is divided in 3 groups of 2 crew members, and every 2 months we have a new exercise which is different for each of the 3 groups. So right now we are using the expanders which are here, the Galileo vibrating plate which is just there, and an MDS which is how would you name that?
It's a multi-purpose exercise machine.
Exactly, the same.
Can do every type of exercise there.
So, for me, for the last 2 months, because it's nearly the end of this period, I was doing the expanders. The idea is to have exercises which are possible to do into space. So instead of doing pull-ups or that kind of exercise directly, we use expanders, which are just bungee ropes. And we can do a lot of exercise by just, for example, for the head, having our hands blocked like that, and then you can do exercise. And so if you want it easier, you come closer. If you want it harder, you go further. And nearly all the muscles of our body can be worked with this method. At the end of each period, of course, we have some exercises which are running or jumping on a plate, which will characterize our power, our muscle gain or loss, how our body evolve throughout those 520 days. So here is a short example of what we do here in the Mars 500 facility.
This is the Galileo vibrating plate from the European Space Agency, and in here we do exercise close to half an hour a day, and we do squats. And thanks to the vibrating of this plate, the effect is much higher on our muscles, and in space this could help prevent bone loss as well.
Some greens inside our spaceship, because first psychologically it's quite nice to see that, and we can even eat some of those plants. So here is the biggest greenhouse that we have in our modules, and you have 12 small pots on which we grow different plants, and I'm sure you can recognize tomatoes here. These are the biggest we ever had since the start of our trip, so we are all waiting eagerly for those tomatoes to become red. We have also radishes, salad, sweet pepper here that we can see, and also peas, which are growing quite quickly. They were put like last week or 2 weeks ago, and onions in the back.
Here in this corner I got some of the sprouts that I also planted at the MDRS in Utah. So there's a little bit corner of MDRS here.
And it gives nice dishes. And here we just began to prepare this experiment, which is also an operational experiment, which is also a greenhouse, and it's called the salad machine, because it's a machine which grows salad. Very difficult for us to know that. So here on the inside you can see a lot of different plates with a number each time, and every 3 days we'll seed salad here in the middle. 3 days later we'll seed salad here, and this axis will move downward, then stop and go upward, and so the plants will think that they are in weightlessness. And at the back, I don't know if you can see, but the walls of this cylinder are covered with LEDs, small lights, and this will replicate the light that those plants could have outside. And so this experiment, I think, will be quite nice to see the biggest salad on the earliest plate, and then going down, see the different growth, different speed of growing for those salads. So this is the latest, I think this is the latest experiment that we are launching in our Mars 500 modules. So I hope that you enjoy this brief tour of our Mars 500 facilities.
We wish you a very productive 14th annual conference of the Mars Society, and that a lot of good ideas will come forward as always, and that we will be able to put them into practice to further the exploration of the red planet, and go there as soon as we can.
Goodbye!
That was so awesome. Just very honored to be able to collaborate them and work with them, and also be invited to continue that relationship with them. And that's what I like to work on, is relationships and collaborations. Because as part of a point of contact for MDRS at NASA, we're also going to be starting and got approved to begin a Mars 101 course, probably a shorter 1-week course at MDRS, to have a more structured field science course for those who might not have a science background or a technical or engineering background, but might want to just experience what it's like to simulate living and being on Mars. And hopefully they'll have a field guide out there to show them the way. And that's also a collaboration that we're starting with the International Space Station and ESA, as well as NASA. So I also have another video, but I want to ask you first if you want me to show it to you. Being with Dr. Chris McKay, he's also working on the MSL, and he was showing an MSL landing video. So I have that. Do you want to see that? Yes. Okay. Okay. There's a little bit of issues with sound in the beginning. It won't have sound then, so we'll see if it works without the sound. You know what? This is where the glitch was. Can we go forward a little bit? So, okay, we're gonna try one more time. If you could just go forward a little bit to keep going a little forward. Yeah. Right there. Can you play from? Yeah, I know. Okay. Okay, here we go. That's not a noise from the MSL. Yeah. Okay. I think it's going to demonstrate the laser, the chem-cam. So right there, it was looking for a good place to get a sample from to actually drill. It's down. No. It just has a couple of sound glitches, and I think the sound glitches are preventing the video. I'm just going to forward it a little bit more, and it might continue. Yeah. Wasn't Dr. Gibson talking about Martian poop? So that's. One can hope. Okay, so that's that. Oh, okay, before you go, so we are gonna, you guys have about 20 extra minutes for lunch, it looks like. But I wanted to let you know of a couple of changes on the schedule. So I'll give you a moment to get that out. So if you look at, this is for the track sessions, since that's what's going to happen next after lunch. For Saturday's track sessions, unfortunately, one group was not able to make it. I think they're in India and weren't able to get here. And that's just track 3 at 3:30. That's with Jaju is how I want to pronounce it. I'm not sure if I'm doing that correctly. That's the only one, the only track session that was canceled. However, we're adding a track session. So in this room here, we're going to actually add track